to the Channel Islands Restoration Environmental Educators Series. Please take a seat, as we say each month. Thank you for joining us tonight for the next installment of our Environmental Educator Series. Tonight we have Kevin Alison of the Catalina Island Conservancy. Very excited. We'll wait just a few more minutes here for people to hop on, and then we will start our presentation. If you're new to our webinar series, your audio and video are muted. But if you have any questions during the presentation, you can submit them to the Q&A at the bottom of your screen, and we will have a 20-minute question and answer session with Kevin at the end of the presentation. Please get comfortable and enjoy the next hour. So I'm going to get started here. We'll do a brief introduction for Channel Islands Restoration. If you've not joined us before, let's see if my screen works here. So we are Channel Islands Restoration. We're a 501c3 environmental nonprofit organization. We do habitat restoration on the Channel Islands and throughout the mainland Central Coast. We also do environmental education and service learning opportunities, volunteer opportunities that I'm sure many of you have attended. And we are also an environmental consultant and contractor. We've been doing this webinar series for a couple of years now, and you can find our extensive list of webinars at cirweb.org/webinars. Look them all go by there. It's a lot. Let's see here. That's the URL. This talk will be up next week as well. If you'd like to rewatch or need to leave early, you can view it there. We would like to acknowledge that a lot of us live in the beautiful homeland of the Chumash people that CIR works closely with on a number of projects. And we want to honor the original inhabitants of this land. That is it for us. Now I will introduce Kevin. Kevin is the Rare Plant Ecologist for the Catalina Island Conservancy. He uses his career and expertise in native plant horticulture, conservation, and ecology to ensure that rare plants of Catalina Island do not go extinct. And as a fun fact for tonight, Kevin is reporting from Catalina Island. So while his background is a picture, he is actually based on Catalina Island, which is very exciting. So I will let Kevin take it from here. And if you have any questions during the talk, please submit them at the bottom of your screen. So Kevin, do you want to go ahead and share your screen now?
All right, sounds good. Okay, just going to, let's see, share there. Can everyone see that okay? I think we should be fine. Let me go to presenter view. Hold on one second. Okay, I think
Looks
we
good.
have it good, right? All right. Okay, well, welcome everyone. Thank you for the opportunity to speak today to you all. We're going to talk today about one aspect of my job. As Maurice mentioned, I'm the Rare Plant Ecologist for the island of Catalina Island. And this talk specifically goes towards a tool or a method of propagating plants to overcome a lot of challenges that traditional methods of propagation can lead to. So my job as the rare plant ecologist is to make sure that our rare plant populations here on Catalina don't go extinct. They're in good shape, we know where they are, and we can be effective stewards of that. And so one way we want to propagate plants is so that we have a stock of them and that we can see that they do not go extinct, that we don't lose individuals, and we're able to capture a lot of the genetics of the gene pool of these particular plants. So a little overview of this talk specifically, I'm going to go over that method I described. So this is plant tissue culture. It's also known as micropropagation. And this method is growing plants in test tubes. And I'll go over the different steps and the methods on how to grow these plants, and then the reason that we're using them as the benefits to these rare plants themselves. So if you haven't been to Catalina Island, it's one of the most accessible of all the other Channel Islands. There are a town in which, like I live in here on Avalon, and we have over 1 million visitors each year, so that poses a lot of challenge as well. But the island itself, Santa Catalina Island, or also known as Pimu or Pamugna by the indigenous peoples on the island, has a long history of visitation and stewardship here as well. And overall, it's a pretty large island, so about 76 square miles, and the conservancy itself manages about 43,000 acres, I think, of that. So a large portion, about, I think it's 88%, so is designated towards conservation, which is excellent. We have over 400 native plants. There is a flora book that should be out this year at some point that will just detail a lot of the natives and non-native species here on Catalina. We have about over 160 rare plants on the island, and we had 9, although that number is likely to change soon, endemic plants, so plants that occur on Catalina Island and nowhere else in the world. I say soon to be because we're still doing some genetic work on a lot of them that we kind of have a good feeling, and botanists think pretty certain that these plants are distinct from a lot of the other within their distribution. Okay, so what is plant tissue culture? And it's also called in vitro propagation, and that's because it is a method of growing plants in test tubes. So in vitro means in glass. So if you were to grow things in a petri dish or in a test tube or a cultural jar, that's all in vitro propagation. It's also called micropropagation because a lot of the techniques we use, like in a greenhouse, sowing seeds or taking cuttings, which is a method of cloning, we do this on a very small scale. So it's called micropropagation. And I'll get into a little bit more about that later, but it is a clonal method traditionally, and it has a lot of potential for really high outputs and really high quality plants. And it also allows you in this really controlled environment where you're allowed to put nutrients and gels and all these and hormones and anything a plant uses in this growing media, in this gel here, we can overcome a lot of propagation challenges. So the methods on how to do this, you can kind of do 2 different ways, and you can go with micro cuttings. So if anyone has done greenhouse work before or has done horticulture and has taken a cutting from something and was able to dip a little root hormone in it and make it grow and produce a whole new set of roots, you've created a clone, but you've created a whole new individual plant as well. And that method still is how we do this in a tissue culture lab, but we're on this small scale, the little bit of growth we get, we can actually take cuttings off of cuttings and be able to exponentially create a lot more plants really quickly. Then there's another method called callus culture. And this is actually, if you've ever seen like a callus or like a mass of cells grow on a plant before, either after wounding or something like that, a callus forms, and you can actually create new plants from that. And I'll get into that a little bit later. So the first step, or the first what we call stage, is called stage 0, or this is actually selecting your plant and preparing that for then bringing it into the lab. So we call it stage 0 because you're not quite doing the lab work yet, but it's the first step. And so this is what we do, where you take a cutting. So you take a cutting from a plant, and you actually are getting it down to just the one little growing point, or the node there, which is much smaller than a actual cutting you would do in a greenhouse, where you would need a lot more material, because when you do it on this really small scale, that plant is going to dry out really quickly in a greenhouse or in a nursery, and it won't have a chance to root before you're able to actually get that plant. So, but in a lab, we can. And so you can see here. Oh, and once you do get the cutting down to that little spot, and or into a little cutting, and get that in vitro, that is now called an explant. It's just terminology. And you can see here, so here's a mechanical pencil next to it. And you can see these cuttings I'm taking are really small. And so the material here is from one little, actually like one or two little sprigs of a plant, which could be only 2 or 3 cuttings. Here I have actual about, I think 30 or so, I think this is like 36 when I do a jar or a rack of cuttings. So I can get a lot more plants to grow in these test tubes just from the start, which other means, or the traditional means of taking cuttings, would take a lot more material, only get about 3 cuttings or so. So it's much more, or much less damage to the plant itself, which is really important when we're dealing with rare plants. Because we're growing these plants in vitro or in glass, and in this, we need to create a sterile environment. So all the tools and the jars and gel is sterilized previously in an autoclave or a pressure cooker. But I need to clean the plants themselves as well, because one little microbe, one little spore of a fungus or bacteria could actually overtake the entire test tube. And because it has everything, the light and sugars and things and nutrients that the plant needs, will also feed those types of microorganisms. So we need to clean them and create a completely sterile plant, which actually also leads to a pretty healthy plant itself. So we need to use a chlorine solution, typically bleach, and this is actually a challenging step, because you have to kill everything, the microbes, but not kill the plant itself. And you need to find the right concentration of that bleach, and the duration of how long you expose that plant to this cleaning solution. And then when you do that, you have to do it in this working environment, because when you open the jar, you're going to have microbes in the air. So we work under what we call a laminar flow hood, which is what we have here. This is actually in our lab. And this is a big blower that blows air through this sterile HEPA filter here, which the pore size on this filter is so small that it actually will catch any bacteria or fungal spores in the air, any dust, anything like that, and just blows a river of sterilized air at you, in which you can actually create like this bubble, almost a sterile bubble, where you can open the jar and you're not have to worry about those types of things, of the contaminations getting in there, as long as you're using proper sterile technique. And so here we are. This is one of our rarest plants, and we are cleaning that in a sterilized solution with a little bit of a surfactant. And I do that for a certain amount of time, and then I rinse them off by gradually getting them in this sterilized water bath after the fact. Once these plants are then clean, I can then open those jars and what we call inoculate, or actually introducing those cuttings into this really brand new environment for it. But think of them as almost like these perfectly clean, sterile greenhouses, of which they have everything they need. Perfect climate, perfect lighting, and just all the right amount of nutrients and sugars and everything a plant would need to grow and reproduce. So once you have the plant in vitro, then you're able to grow them out. And so the next stage would be to multiply them. And so you can see here, this is a manzanita where I was able to initiate a whole bunch of new shoots. And then from that, I can actually take these cuttings from the cuttings. And so I can multiply quite a bit of clones of these plants using this method. And so in a growing season, this might take, you'd only have kind of 1 or 2 points where you can really take cuttings off of a plant. But in the lab, you can do this almost year round with complete climate control. And so you can rapidly, exponentially clone these plants. So let's say I have one plant and I can add a shoot hormone. So we have root hormones. We also have a class of shoot hormones. And you can find what the plant needs, and you can actually stimulate and initiate new plants to grow. And so let's say I get 10 new plants to start to grow from that mass. Divide them out, I have 10 new individuals. From those 10, I use that same multiplying media with those hormones in there. I can get 10 off of those, and I can quickly get to 100 new individuals. With that replication over and over, you can get to potentially 1 million plants in just 6 replications. And this is important because when we work with rare plants, the amount of times we're able to clone a particular individual is, especially for in a low population, rare plants, there's a chance for every one of those individuals contributing to the gene pool to not be lost. And so we want to have these replications so that if in the wild, one plant kind of dies, we can replace it and make sure that those genes are not lost. The other method is callus tissue. So this is a lot what I did with my master's work, working with Dudleya species. And this method of propagation is really new for us. We're on the kind of ground floor of this. And so a lot of the photos are from this method is through my grad's work, but we're going to be applying to some of our rare species here. So you can see when I said it looked like a mass of cells or a massive tissue, it's literally that. It's this green, initiated by root hormones and things like that, or a combination of hormones. Think of it like stem cells. It's a mass of cells, but they're dedifferentiated, meaning that they're not a shoot, a leaf, cell, a tissue. It's none of that. It's actually dedifferentiated. It's just a plant cell. And plants naturally do this in different responses for particular reasons. But we're able to capitalize on that because after the fact, I can then switch the concentration and the type of hormone to then every cell in this mass has the potential to be a brand new plant. And so that's incredible. So we did that with the Dudleya, and from that I switched to a new media, and we're able to create new plant organs or complete new plants from that mass. And so that's called organogenesis. You're creating new organs. And so for here, you have the Dudleya on the right. You're actually seeing quite a bit of new Dudleya forming. And then you have to kind of get the concentration right, because you can see on the right here, roots started forming when I was initially wanting shoots first, and so I could divide out those individuals that you see here. So I have a whole mass of cells. I have a whole bunch of new plants. So I have way more than the 10 I was originally shooting for with this, but then I can divide those out. And quickly, I have a whole bunch of new plants and new clones. Now, why I did this with Dudleya at the time was because we were trying to flood the market with really rare plants. so that the horticultural trade, to combat that. I don't know why it keeps switching on its own. But we were trying to combat that, the poaching that was happening throughout the state of California and in Baja. So we were trying to get ahead of it and flood the market so that we can decrease that poaching pressure. So that's where this method became a really important tool for conservation, by supplying these rare plants. So once you have a complete plant, you can then switch that media to a root hormone. And you're really in the driver's seat with this type of method by whatever you want to put in the media towards mimicking what the plant would do. So hormones are a class of chemicals that are signaling compounds. So and that's how we do it in our own bodies without us knowing. And so do plants, where they, through these concentration gradients within the body of the plant or ourselves, things happen, and it signals to do something. So when if we dip a cutting and we want new roots, that root hormone is signaling to growing points in the plant to then produce new roots. And so we can do the same thing here in the lab too. And so once we've initiated new shoots with shoot hormones, can divide those out and then root them out using root hormones. But you can also sometimes skip that step if you want to do that out in the greenhouse, but that really depends on the plant. So that's what we did here with that Dudley. And then if you've ever done greenhouse work, you have to toughen up those plants because they've been in such a really we've been babying them. So their tissue cells and everything that they developed were in a really nice environment, really relaxed for them to do a lot of growth, which is great and what you want. But you need to expose them and gradually to an outside environment where they eventually will live. So you want to gradually decrease the humidity and toughen them up, or what we called hardening off these plants. And that you would do that in a greenhouse, like if you were and you're then exposing it out into a growing yard. You might do that with some shade cloth, and then eventually wean them off until they're ready and they can build up new cuticle layers on the leaves and thicker cells on those plants themselves, and they're able to then, or thicker tissues, I should say, to then be able to handle growing out on restoration jobs or anything like that. Okay, so how this applies to our really rare plants, there's two main species that we're using this technique for, really eventually three, but I think the two are the main ones. So we here on Catalina have the Catalina Island Mountain Mahogany, or Cercocarpus trasky, and that's here on the left. Now this plant is really in a lot of danger. So this is actually a federally endangered plant, state and federally endangered listing. It occurs only in one spot on the backside of the island, and we only have 6 individuals left in existence. And so a lot of people call this the rarest tree or the rarest shrub in all of North America because we have that. We don't include Hawaii because they're not part of that North American continent, and they have a lot of rare plants there. But here we have this extremely rare plant, and it has a lot of challenges. It's really a tough place where it lives in Wild Boar Gully, and growing this plant and its reproduction is really difficult. So in the wild, this plant has basically lost its ability to make pure seed or to make seed of that particular species because it's surrounded by this species on the right, the Cercocarpus betuloides, which there's betuloides all over in California, on the mainland California. We have a subspecies blanchii that actually occurs on the islands, but it's the most common that you would find on all the islands, really. But the problem, or why I say that the Cercocarpus trasky, or our endemic, the rare one, has lost its ability to make seed, is because it's the pollen pressure on these 6 individuals is so great that basically all the seedlings that come up are hybrid. And that's not a great thing when this plant is basically being bred out of existence. And the further, and once that those hybrids on top of the other ones, and once the hybrids hybridize with the 6 individuals, you actually, these individuals can get further and further away from actually being Cercocarpus trasciis. So it's a phenomenon called genetic swamping, and it's really challenging for a species here where there's only 6 left, so that there's such a small population size. So what would you do with this? So like I said, it's extremely rare plant. It has that genetic problem, genetic swamping, where we can't collect seed and put it out or use it for restoration or for gene seed banking or anything like that. So we have all hybrid seed, and the cutting material, although not impossible, is extremely unreliable, and they just do not take. We've tried many methods, and they just don't. And so you don't have a reliable method to capture the genetics of that plant. And so if one dies of those 6, that's a healthy population genetic lost completely from the pool. So it's very, very drastic, and a problem we need to overcome to be able to conserve this species. So what do? You basically lost your ability to make seed. You've lost your ability to do cuttings, and that's where plant tissue culture can be very valuable. So this is what we're doing now. We've just started this, so you guys are getting a sneak peek into the hardcore science that's going on at the Conservancy. So we've established a lab, and this is one of the parts that I'm really working on. And so I got to develop this protocol. I have to find all the right concentration of sterilizing solution, the bleach, and so I don't kill the plants, but also kill all the microbes, see how long they need to be exposed to that, get it through that sterilizing hurdle, and then find the right combination of macro and micronutrients and all those things that the plant need to grow. I have to find it by observing, and this is horticulture, this is propagation, and using science to figure it out. And the plants will tell you. They'll tell you what they want when they're healthy or not. And so we have those signs. That's what I'm working on. So right now we've been able to establish cuttings in nice, healthy, clean plants. So we've gotten through that step. But the problem I'm having is I'm getting that callus culture. And it's like, great. But what's happening is this is happening without any additional hormones being added to the media. And so that's really concerning. It's really, okay, well, why is that? But what happens is it distorts the tissue so much and kind of goes on this runaway effect where it quickly will self-oxidize and die out. So although that method of producing the callus, which is a method to grow plants, we're not getting healthy callus tissue. And so it's a really difficult method to grow plants. So it's a problem. So I'm getting unwanted callus. That callus formation is actually a stress response. And so the plant has a lot of internal, or what we call endogenous hormones itself, that's causing a lot of this callus. Ideally, I would like to be able to do the other route where I have new shoots, they grow up, chop, 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 and I have plenty of new cuttings and individuals that I can save. The next thing I'd like to do is test the other method, but try to create healthy tissue. But in order to do that, I wanted to prevent that callus. And so what we did is there's a anti-auxin, which is a group of hormones. And we had just recently trialed it, and it worked. So here's a brand new cutting. A few weeks later, healthy, you don't have that callus. So great. It's a lot of science. We had to trial the right amount of concentration of that particular type of, it's basically a hormone blocker of a specific type of hormone that I had assumed was making that callus. And that's where we're at. So now we have healthy new plants established in the greenhouse or in the lab, which is actually the most important step. Because once you have these plants established, now you have it basically a repository, or it's saved in vitro. And I'll talk about that in a little bit, of why just having them in the lab is so important. So from here, we're going to try different class of hormones, and we will start to initiate new shoots and hopefully create whole new plants once we have roots, and then we have all these healthy new clones. The other plant we want to work with is the Catalina Island ironwood. So this is an endemic species. There are other island ironwoods of the same genus species, but this is a subspecies that occurs here and only here on Catalina Island. And it's a beautiful tree, and historically it produces these suckers. It's not really producing seed, which is interesting, and we're trying to figure out just exactly why. But what happens is we find out that all these plants are actually, or all these trees in these groves are actually clones of each other. And we have about 120 groves on the island, and we found that each individual in that grove is a clone, but every grove is distinct. So we don't see much seedling recruitment or anything like that. So they're not producing really viable seed. But when we have 120 groves around the island, which seems like thousands of plants, we actually only have basically 120 individuals, genetically speaking. And so you see that they see these little basal sprouts come up from the tree, and you'll start to get stands of those basal sprouts, and they separate over time. As you can see, logs start to die out. So here you have this distance where you see all these dead and sprouts or what appear to be individual trees have died out, and you basically have separated out further and further clones, but they're all genetically identical. And so it's just really just one big individual plant that's really, really old. And so that's the next step, is we're trying to get those plants in every grove represented in the greenhouse. Again, it's another plant that does not do well from cuttings. And so we're trying to establish them in the lab. And then we'll have a repository of basically, I can have the entire genetics in every single grove growing on a small shelf in a lab. And then I can just, okay, this particular grove, we need to do restoration, we need more plants, or we need new cover just to create the habitat there for everything else, and also preserve those plants. I can do so. We can grab it, start to multiply them. We'll grow them out eventually in the greenhouse once they're ready, and out back onto the landscape. So why I say that's so important, and I know I usually get this question about, like, well, what isn't genetic diversity such an important part of this? Absolutely. But what we're trying to do is collect the clones of each, when we get to such small population sizes, instead of collecting seed, because then we capture the entire genetic pool, and then we can create groves and then do cultivation and that amplification of genetics later, the recombination through sexual reproduction, and use seeds from there on out. But right now we want to make sure that we get clones established, because this is the most important step. We can either go towards production, where we create new individuals. We can get these out on the landscape and create these conservation groves, where we basically reproduced, like in the instance of the mountain mahogany, we can create that grove elsewhere on the island where there's not that hybrid pressure, and then let the plant start to naturally reproduce through sexual means and produce viable pure seed out back on the landscape. And same thing with the ironwoods. Maybe that could also be the case. Maybe the problem we have is that through climate changes, things here, maybe the groves are too far away from each other. And so maybe we need to find a way where they can start to then be able to reproduce naturally. Maybe there's a pollinator that went extinct. That's really the case we see a lot in Hawaii. That could be occurring here as well. But either way, we want to capture that genetics and make sure that it doesn't go extinct. So keeping them nice and healthy and safe in a lab in these perfect growing conditions is essential. And so just like the way that we store seeds, and for what we call seed banking, we can do the same thing with living plants in a lab. And so that's a method called germplasm storage. Additionally, tissue culture and growing these plants sterile in a lab is the intermediate step towards cryopreservation. So we will be working with the San Diego Zoo that's creating this frozen zoo where they're going to be doing rhino embryos and things like that, and cryopreserving them or freezing them, and so they can reanimate them if there ever was a problem with a plant where they go extinct. They want to do the same thing with plants, and so we have this opportunity where we can get these 6 individuals left of the mahogany into that frozen zoo as well. And then hopefully that is a method where we have basically saved the plant from extinction indefinitely. So this method of plant tissue culture, that, like I said, it's very minimal pressure on the plant. In the wild, we can collect just very small amounts of tissue and be able to grow and keep clones of it. It's a rapid method of propagation. We don't have seasonality as much. We have perfect growing conditions, and we can overcome that and take cuttings and cuttings very quickly through that exponential replication. It's also biosecurity. When we grow these plants, we know these plants are clean, healthy, and free of bacteria and fungus and phytophthora. And we know that these plants are clean and sterile. So it's a biosecurity issue too. We can grow plants for, once we develop this protocol with the ironwoods, we could also do the Channel Island, the Northern Islands, or their subspecies there, and we could grow and ship them. And we know that those individuals we're sending are free of weed seeds. They're free of any pests that may start to would be a problem elsewhere. And then other methods that we have this, you can do embryo rescue. So let's say we have an acorn and we have a virus that's attacking acorns or the trees, and we're not getting by. We could actually excise, or say we have a pest or something like that. We could excise and save and remove the embryo from a seedling, and it can still place them into this perfect growing environment of a test tube, and grow out those embryos and grow those seeds. That would be nearly impossible in a greenhouse or a nursery, because it's just too damaging to the plant. And so yeah, so it's basically like a baby that's premature. We're able to provide that type of hospitalization or that type of environment where we can baby them through and grow them into a healthy plant. We can also create synthetic seeds. This blows my mind when I first heard about this. So with the embryos that we excise, or when we do small little plants, we can actually encapsulate them in a liquid gel or seed that dissolves. So we take like liquid gels, it's basically the same material. But you can actually have a little plant in there that has like pre-packaged with fertilizer and everything in that, where you can create basically a synthetic seed. It's basically, instead of it's asexually produced, you have a clone. So like these 6 individuals, we could create these seeds and keep them in a cold storage for years, hypothetically, and then be able to grow these out this way. So this is another method and tools that tissue culture allows. Virus eradication is another method. Virus doesn't attack the very fast, quick growing environment of the meristem. We could also excise just that really quick growing point and grow those out in the lab, and which would be too small for a greenhouse or traditional method. But we can actually basically physically, not chemically, physically remove viruses from plants. So if they're God forbid there's anything like this that happened on our rare plants, we would be proactive in a way to be able to overcome that. So the tissue culture lab and this method, I like to think of it as another tool in our conservation tool belt. So we have the nursery to grow seeds for restoration, and we have the tissue culture lab to respond when those plants aren't able to be reproduced or grown through traditional means. The Conservancy is also working towards developing a seed farm. So we're going to be able to amplify a lot of our seeds here and be able to reestablish and enhance and supplement our seed banks back on the landscape to be able to do large scale restoration as well. Very excited about that. And then our botanic garden, we can actually keep a repository, what we call ex situ conservation, where we have a place where we can grow these plants outside of its normal environment, but have them also growing in a garden for everybody to enjoy that may not be able to get out in the landscape and can learn about it, which education is incredibly a part of conservation, which you guys are all here today to do. But also it's a way that we can actually go back and learn about these plants and have a safe keeping where we need to take cuttings or anything if it gets back to that kind of situation as well. So yeah, so with all that, that's the presentation, and I will go back to Maureen.
Yeah, thank you so much, Kevin, for the presentation. Your organization is doing wonderful work out on Catalina Islands regarding plant propagation, and a lot of interesting material in your presentation. So I think we have a few questions now. Let me pull up the Q&A here. Let's see. Nicoletta wants to know, how long do plants survive in vitro? How reliable is something like germplasm storage compared to seed banking?
Yeah, so great question. The best part about that, so I've had manzanitas before when I've done tissue culture with that, and I've been able to keep it nice, healthy for about a year without even touching it. And so on that growing media or anything like that, you can really keep it in a juvenile state in this really small environment or growing environment. So there might be a little bit of research on that about how long it actually will impact the plant itself, but we're keeping it at a real small environment or small stage prevents and keeps it living really well. And if you think about like in a greenhouse with a grown and living plant, you have to be there watering it, you got to be there feeding it, make sure it's all its nutrients are, it doesn't have nutrient deficiencies and pests and all these types of effort that goes into it. Once it's clean and living in vitro, it can last quite a long time. And so I like to call my test tube babies. You then can go in and we basically change its diaper. You just change the media and put on fresh new media, and they respond really well in that way. Compared to seeds, that all depends on the plant. So some seeds are only viable for a certain amount of time. We do seed banking and keeping them in cooler environments to be able to extend that. But in both cases, they are they're living things that have a lifespan. And so a seed actually is just a little living embryo encased in an environment under suspended animation. So there is, at some point, those seeds have to be germinated. The problem with seed banking, though, is that you can lose viability and then those seeds won't germinate. Here, we have plants already germinated and established or growing that we can then move to the next step and then get them out living. So, it also kind of overcomes some challenges we have with seed banking as well. And so, yeah, so I hope that kind of answers your question. How does it compare? That all is plants dependent. But the process is there's benefits to both. There's pros and cons to both sides, but there's a great amount of benefit as well with plant tissue culture.
Thank you. And sort of a follow-up to that, though maybe you just answered it and sort of it's dependent, but a couple of people had questions that, plants that are raised in sterile environments wouldn't be as resistant once they're planted out. How do these plants compare when they're in an outdoor environment as opposed to in the lab?
Yeah, so that's an assumption. We don't know. We have to test that. But we do use, but as the tissue culture and growing point, start is just one step. So once we do get them into a greenhouse or get them growing out of the lab and then into the next step, which is growing them out into containers that you would plant then onto the landscape, we do a lot of that type of things where we would use beneficial organisms. Sometimes we take duff, especially in the cases with oaks, and inoculate it just like we would, with probiotics in our own type of cut, or like, how we get those organisms are really important as well. So they are sterile, yes, but it's one step, and that would go with seedlings as well, in a traditional method. So the plants actually will respond pretty well with that, not having the bacteria and all the microorganisms and primarily fungal and other things like that, but they can quickly be inoculated with those good type of things and grow out. So we haven't seen that really be a problem with not being or coming from a sterile environment to then the non-sterile environment. It is a challenge in that step, but it's another part of that hardening off process. But as long as you have healthy, good plants, they seem just growing wise where their structures and everything are pretty good. They can transition well to then being having those microorganism loads, and especially with the good ones we want, and start to live and proceed from there.
Yeah, thank you. And carrying on that train of thought, both Terry and Robin had sort of related questions. Terry wants to know, can the clones be remixed can the genetic material of clones be remixed, occur with sexual seed production? And Robin wants to know if the abundant number of native plants for landscaping, is this a viable option to meet those requirements? Or would this be a viable option?
Yeah, can you repeat the first part of that question?
How can the genetic material of the 6 clones, can they be mixed in with sexual seed production?
Oh, okay. So we're talking about the mahogany with those 6. Okay. Yeah. So I'm not mixing any genetics. I'm not doing genetic manipulation. I'm not playing with the DNA or anything like that to make these clones. But what we're doing is creating, by just like how you would take a cutting off of a plant, we're doing this at a small scale in the lab, and we're creating clones of each of those individuals, and then yes, later we will then place those on the landscape in what we're calling conservation groves, where we've replicated and we'll plant like each one of those 6 elsewhere on the landscape where they can do that naturally. So we're just removing that pressure of the hybrids and having other opportunities, and not all of our eggs in one basket in Wild Boar Gully where these plants occur, where there's a landslide that basically came in and already have almost killed one of those 6 individuals. And its root balls exposed, and it's a whole mess. But yeah, I'm not manipulating the genetics at all in the lab. This tool is used though for genetic manipulation. We're just not doing that. We're just growing the plants. But yes, there is a method where you can take and manipulate genes and using this method, but that's not what we're doing. That's a whole other tool and science part of it I don't even want to get into.
Great, thank you. Let's see
Oh, to
here.
answer the question about the horticultural trade, yes. So that's what I was doing with the Dudleya, where we were trying to get preemptively, getting rapidly through using that exponential clones to flood the market with Dudleya. So you may actually see a lot more Dudleya out on the trade, especially from Tree of Life, where I was working before this. And those rare plants are available and can be produced and will crush that poaching pressure. So it's access was the problem. There was a economic problem, really. And so you have cultivars. So these are plants that have traits that would do better in a garden setting. That's one species. You have hybrids as well that do in function, but still produce a ton, if not even a lot more, and in some cases, pollen and nectar for certain plants by extending the amount of flowers that a plant might have. That's attractive, but can also be a big provider of resources for native pollinators. And so tissue culture allows you to clone, and cuttings as well, but tissue culture can do it on a larger scale to supply the horticultural trade with these native plants. And that's what I was kind of working on before when I had the lab at a native plant nursery. So yes, it can be a big benefit for the horticultural trade, especially with native plants.
Great, amazing. Robert has a question specifically about the Traschia, but he wants to know if there are certain limitations for soil types or elevation that would impact where you can plant the nursery plants.
Yes, so my neighbor here and our wonderful GIS manager has helped us to be able to develop these, what we call species habitat models. So we're able to use the traits and soil types and elevation and aspect, the slope in which they occur, to be able to find areas on the landscape or on the island that are good potential that checks all these boxes for us, so that would be recreating that habitat. So yes, that is something we look into when we select these groves.
Right. A couple people had questions on specifically how much solution, how long to soak the cuttings, where they can find information about those specific processes.
Yes. Oh, you know what? I forgot to grab it with me, but there is a book that's called Growing Plants from Test Tubes, and it's actually where I got some of the slides here. Let me see. Here, I can pull it up real quick. So specifically what concentration, everything like that, that is all species specific. So that depends, every plant will be different. The challenge we have is this has never been done before with these species. So this is something we have to figure out. Generally speaking, a concentration of about 10% bleach to water for about 10-15 minutes can work. But the Cercocarpus traskii, or the mountain mahogany, has a lot of really fuzzy hairs. That's part of why it's so distinct from the other species of the island mahogany, or the Cercocarpus blanchii, or Petulias blanchii. And so that actually, having all those hairs, has a lot more surface area for places of bacteria and fungus to hide, and for water, that's cleaning solution to actually get in and around the plant. So you have to have either a higher and longer concentration, or higher concentration and longer duration, but still not damage the tissue itself. So it has to be something you do experimentally with using science. And so trying to be able to find that can be a challenge. A challenge, but also a fun puzzle, I like to say, to try and figure out. If anybody wants to learn more about tissue culture itself, There's a really good book that really will let you and get you what you need to start. And you can start doing this in a home lab, creating a sterile environment. So I'm going to share my screen real quick so you guys can see this. Yeah, I'll just do it from there. So this, you guys see that plants from test tubes. This was actually my textbook when I first was learning how to do this method. If anybody wants to screenshot that, I can also put a link to it in the chat. I am not affiliated with this book at all. It's just an excellent, well-written research or a well-written book that is one of the textbooks. I think the best one out there as far as which one to, or how to do this plant tissue culture. And it'll go over all that about the different processes and how to go about and approach the challenges of trying to create the media and each step of propagation and things like that.
Right.
Okay.
Yeah, a couple people asked where they can get more experience about this, since it seems like a fairly new field. Someone specifically mentioned where people can contact you about embryo rescue. So maybe now would be a good time to share that if you have that information.
Yeah. So as far as how to learn more about this, I think that book does a great job. As far as embryo rescue, so that's something we haven't done yet here. We don't, and luckily we haven't had that case. Where to learn how to do this? Oh, that's a tough one, because that was the challenge I had when I tried to learn how to do this. And I got a degree in horticulture, I got a degree in conservation, and I was trying to match those two once I found out. And so I actually went to the University of Hawaii at Hilo that was offering this as a certificate program. Unfortunately, I heard that they no longer offer that. I think the main professor I learned from, Dr. Michael Tanabe, retired, and so he was kind of the one, the guru to learn how to do this. And so I was lucky to have that opportunity. Some community colleges and horticulture might offer it as far as, and universities might have it as like a unit in there. I would contact them and see, but I don't know who's teaching this out as an actual skill set.
Yeah, no problem. As a follow-up from that, I had several questions. If there's other organizations doing similar work that you know of?
The San Diego Zoo, I know, is doing a lot of work with this with Quercus dumosa. And so it's great. We've been working with them. Joseph Ree, he's a postdoc down there, is doing a lot of that type of work. And he's trying to do a lot of different oaks. And luckily, he trialed it with Quercus tomentella, which is our island oak. He did it with the ones on the Santa Cruz Island, or Northern Islands. I think it was primarily from Santa Cruz. But he was saying that it's doing that Quercus tomentella was one of the best that responded well to in vitro. So we're going to then use it for our oaks here on Catalina, which we have. It's one of our rarest oaks as well, is Quercus tomentella. So yeah, he's done an amazing work and has put, you know, all that effort and years into developing a protocol for it and the media and everything. And we're going to trial it with ours here pretty soon. There might be a little bit of phenotypic difference, so maybe even just variation between islands. Maybe there's something we have to tweak. We don't know, but yeah, he's, as far as tissue culture in California towards that, I think he's kind of the only one that I know of besides me and him. Like I said, Hawaii is definitely developed using this. The Missouri and Cincinnati Botanic Garden are doing a lot of tissue culture work with it. And folks at the Huntington Botanic Garden, they're also doing plant tissue culture, but I think they're more focused on certain strains of like avocado and food crop things. I don't know how much they're doing California natives or island ones, but I think they are doing some rare plants as well around the world. I think it's some of their cacti and succulents they might be working with as well. But yeah, it's a small group of us working and using this method.
Great, thank you. A couple quick questions here. Anonymous wants to know, how do you get the microplants out of the test tubes?
Very carefully. So, so yeah, it's very delicate. What you would do is you can almost put them. What I like to do is actually first I put it on media where I wean them off of sugar, because sugar, it's why we have sugar in there is they're actually supplying it. It's sucrose. Some plants can use different compounds of that, but literally I could put these plants in the dark even, and they'll still grow and respond because they don't. The sugars are providing what photosynthesis would be, essentially, and well, first photosynthesis and then respiration, to actually use, or sorry, just the photosynthesis, is providing that sugar. So I actually wean them off slowly, and then they're kind of more responsive to then start to use their own, and it's kind of one method to toughen them up. But then also you put them in a situation in containers or other where they get their humidity just incrementally dropped, and so you can do these in little chambers, even like humidomes and things like that. You just make sure that they have enough humidity, and you slowly get them um, uh, hardened off, and so they're able to get used to environment. And it's just an extra step, like you would go from a greenhouse to then out in the growing yard and then out in the landscape. You kind of go lab, greenhouse, growing yard, and out in the landscape. And so, yeah, you just got to do it very carefully. Some plants respond very well. That, like the Dudleya that I was doing, you can go straight from new shoots out into a greenhouse, and they just toughen right up, no problem. They're really tough. But something like mahogany, or especially an herbaceous type of plant, if you work with that, you would have to. It would take a lot more delicacy.
Great, thank you for the answer. Few more quick questions here. When you plant a clone plant, do you have to start a new grove, or can you plant it near the original?
I wouldn't, just because we're at such a small population size. You don't want that inbreeding type of things to happen. Although I don't know, like I don't know how is exactly that would impact the genetics of it. But to err on the side of caution, I at least know that if, like I said, that one landslide came and wiped out tree, I think it's tree D, um, I can replant tree D from a clone, and I have essentially no genetics was lost whatsoever from the gene pool of that population. So that's why I say when we replicate, we have multiple clones on a shelf, that's multiple times that exact individual doesn't die. So imagine if there was a thousand Kevins, and every time I died, I actually was not, I was replaced by one more. That's basically the type of situation we're trying to do with our rare plants. Now we wouldn't breed them because we didn't have that inbreeding that would occur, but that's why I was saying creating them as grove. We've just basically cloned the grove and placed it somewhere else, and there's multiple groves around, to then start the recombination of genes.
Right. We have a couple more questions here. Let's see. I'm going to see if I can read this. So Robert wants to know if the beetle,oids, if they're removed in the area of the trachea, can they be removed in the area of the trachea in order to have no genetic swamping risk? I'm
Yeah.
assuming that,
So
yeah, go ahead.
okay. Yeah. So we have done that. The problem is we still get hybridization. So we don't know the mechanism in which that these plants are starting to do that. So we don't know the distance. We don't want to just keep consistently removing, in a huge buffer. But we have provided a buffer in which those you're talking the Cercocarpus betuloides
Yes, thank you,
plantae,
thank you. Yes.
the island mountain mahogany, which occur on other islands as well. So we have created that, but we still get hybrids. So is it wind blown? Is there just a really effective pollinator that can get all the way around? It's probably a combination of both, but I think that wind pollination and these mahoganies put out a lot of pollen, then is causing that problem. So that's why we say it's basically lost its ability to make seed. We did leave the hybrids though. And although we have the hybrid trees there, and they do back cross with the original 6, and we get F2 hybrids. And that's why I say it gets basically genetically further and further away from being that pure Trasky. That we left those because it's actually a somewhat, if we were to lose tree D and we don't have this protocol developed, we still have a portion of tree D genetics in one of those hybrids. So it is a method of conservation, but it's also, it's kind of pros and cons. It's a little bit of both. But when you're looking at preserving the rare plants, we're preserving the genetics within a population. And that's the money in the bank that makes things go. And so we're trying to preserve that. So we didn't remove the hybrids, but yes, we did remove and create a buffer. The problem is we don't know exactly why, but we're getting a lot still hybrids.
Great. Karen Cotter says, great talk. Thanks for sharing your work. Thank you, Karen and Kevin. Another lengthy question from Robert here. Now he's talking about the ironwood. He wants to know if there's any evidence of deer, bison, or insects causing risk for the clonal roots.
Yes. So bison, it's too steep for them to get to where these groves are. So I haven't seen any issues with bison, but I have seen deer. And previous years, pigs, sheep, and goats were the major cause of habitat destruction and things along those lines for these groves. So there's differences between browsers and grazers, and bison are grazers. So, and the problem we have with ironwoods is the deer are browsers. So they'll actually, all those new shoots coming out, they eat those like crazy. And so our only method at the current for us is exclosures and fencing, but those fences go down. Often we have to constantly maintain them. But yeah, they are a major problem for us. The other thing is that when we do get some seedling growth, there's if we do see seeds come up, which happens very rarely, we find that, yeah, and this goes for all the plants, and especially island endemic plants on Catalina, is that the deer will eat those, and other the ungulates will eat those seedlings. And so the new sprouts and seedlings, the two method that the groves have as far as reproducing, these ancient plants basically they've lost their ability to reproduce because of that. So yeah, it's a big problem.
Okay, and we have one more question for the night. And as a reminder, if you'd like to view this talk, you can find it on our website next week at cirweb.org/webinars. Thank you again, Kevin, for joining us. I'll also mention that next month we have another Catalina Island affiliated webinar that you may have mentioned at the beginning of the talk about the flora of Catalina Island, and that will be with Matthew Gilliams from the Botanic Garden in Santa Barbara. So last question, Peg would like to know if there are effects of fungi, or have there been studies on the effects of fungi on the network of seedlings once they have been planted in natural areas. So if there have been any what studies have been done on the effects that fungi
Seedlings in general, or micro-propagated plants?
I believe she's talking about your micro-propagated plants. Yes.
Oh, okay. So we haven't gotten to that stage, and so we don't know. But yes, I am looking towards an affiliate that could, once we get to that point, it might be that using mycorrhizae. So first we want to see if the plants themselves have a mycorrhizal association. And we know that they do with ironwoods. And I don't know about Cercocarpus, but we should look into that. And yeah, if there is that association, we would like to do that. it's likely a benefit. And so we haven't gotten to that stage for our micropropagation plants, but it is absolutely, you bring up a great point, something worth looking into.
All right. Well,
We
yep.
have seen that with oaks. Oaks have been heavily studied, and so yeah, you can just take kind of a soil around or part of the rhizosphere of the plant where oaks come from, the duff, and mix that into the media, and yeah, you do get. It's been clearly studied that yes, that you do get better establishment, recruitment, and survivability of a lot of plants, and not just oaks. I think that's been studied pretty well, and you do get that effect. So we would do the same with these species and likely see the same result.
Great. Well, thank you again for joining us, Kevin. That was a really interesting presentation. We appreciate all your work that you're doing on Catalina, and we will see everyone next month. So have a great night, and thank you.
Thank you very much.